Allie, you're first up, so you need to be in the batter's circle here. All right, just a couple of announcements, a couple of things before we start. If you have a rental bike and you have not yet returned it, and you rode it to the lecture today, you should return it on the way back to lunch at the bike barn. If you did not remember to bring your checkout sheet and you have a rental bike, then you're going to need to get some sort of written note from the bike barn and staple that to your checkout sheet so that we have some evidence that I am not buying a bicycle with your deposit check. All right? We're going to try and stay pretty close on schedule. You're being, be good audience members. Please turn off your phones, any devices that are going to make noise. Whoever's plugged in over here has to unplug that. We're not allowed to have wires across the aisles. So please unplug that and move the bike helmet and the seat, everything out of the aisle. That can go on one of the back seats if you want. There are no Pokemons in here. We should not be hunting any Pokemons. And I believe this is an assassin-free zone. So, with that, George, are you the only one alive, George? No. I know Henry was alive last night. Are you still alive?
No, actually, I'm not.
You are not. Henry, I'm so sorry. All right, so it is my pleasure to welcome you and the people who are watching from as far away as Greece and South Korea to the presentations for the 2016 Young Scholars Program. And leading us off is Alan Mays.
Hello everyone, my name is Allison Mays and I'm working in the NOS Biology program for measuring and predicting syringe hub loss. The syringe has three parts. It has the barrel, the hub, and the needle. And whenever the hub is there so that the needle can be attached and reattached from the syringe. Whenever the measured liquid is expelled from the syringe, there's always a little bit of liquid left in the hub. That is called the hub loss. This is a conventional syringe. This is a tuberculin syringe. The difference between the two is that a tuberculin syringe does not have a detachable needle, so there's no hub and almost no hub loss. Controlled substances are substances that are monitored by the DEA or the Drug Enforcement Agency, and the reason why they're monitored is because they can be easily misused. So labs and institutions that use animals often use substances like ketamine or buprenorphine. Ketamine is usually used for anesthesia, and buprenorphine is used for pain management. They use drug logs to show that what is going in and out. No, not in. They use drug logs to show what is coming out of the vials and why. But at the end of each drug log, they have to balance how much came out and how much was the starting volume. And because of hub loss, there's always going to be, you're never going to be able to take out how much was originally in the vial. So there's currently, as in the past, there has been really no study focusing on what hub loss should be allowed per vial. So past research has focused on just the syringe hub volume itself. One of them looked at how dilutions are affected. by the hub. So whenever a dilution is mixed, whenever a drug is diluted within a syringe, the hub volume is typically 0.07 mils, which if you're trying to draw up 0.02 mils of the drug, 0.07 is added to that, and then there's an overdose, which is not good. So this is really important. With each syringe, there's some hub loss, so the controlled substances are being wasted and costing money. High dead space syringes waste more substance than low dead space syringes. And dead space is just another word for the hub because the liquid in the hub is not being utilized, so it's called dead space. A high dead space syringe would be like a conventional syringe, when a low dead space syringe would be more like a tuberculin syringe. The applications to this study would be that it's going to help the DEA and other institutions be able to better monitor hub loss so that it will both protect the people who are doing things correctly, because maybe the hub loss is a lot more than one would think, or it can help to see whenever the drugs are being misused. Another thing that we can gain from this study is improving lab techniques so that we can save money by not wasting controlled substances, and we may increase the accuracy of the drug dilutions and injections. Our hypothesis is that the number of syringes would increase the amount of hub loss per vial. We predicted that the data would be fairly consistent and accurate because a syringe is typically seen as a reliable way to measure liquids. So we figured things would be fairly consistent and not very different. The study design, there was two parts to our study. There was the part where we looked at just the hub volume of each syringe, and then there was another part where we looked at how much hub loss there is per vial. We had two variables to the first part of our study where there were 3 people doing the same tests on these different syringes 3 times, 5 times each. The 3 syringe types, this is used in our vivarium court of the mouse biology program. It has a smaller needle and it has a conventional hub. This is a tuberculin syringe. It just does not have a hub because the needle is not detachable. And this is used in our phenotyping core with a larger needle and a conventional, phenotyping core, okay, with a larger needle and a conventional hub. We had 2 parts to this first part where we had a mass test, so that way we could get more accurate measurements. What we would do is we uncap the syringe, weigh it, and then, like the empty syringe, and then we would pull up 0.5 mils of water. After that, we would weigh that, expel the liquid, and then into a waste vial, and then we would weigh the used syringe. The difference between the empty syringe and the used syringe was what we considered the hub volume. The visual test was closer to the way that people traditionally use a syringe, in that we used the measuring diagram on each syringe to tell how much was in the hub. We would draw up with each syringe 0.5 mils of water, expel that, and then pull back the plunger to about 0.1 and see how much had been left in the hub. What we found from this were 2 things. We found that the measurement techniques were comparable. You can see that the difference between the mass test and the visual test was not very significant. So because they were comparable, we were able to safely say that the visual technique of measurement was okay for the second part of our study. The next thing we saw was that interpersonal variation does have a significant difference in the measurements that we get. Each different bar represents a different person's measurements for the same test. The next part of our study we had 2 parts to this as well. We had the half contents test and the entire contents test, but we had issues with the entire contents test, so I am not putting it in the presentation. But the half contents test simulates whenever a drug expires. So what we did was we would only draw out half of each vial, and then from there, using these intervals of withdrawal. So we would use, if the interval is 0.1, then we would draw 0.1 mils with each syringe. And we would use this number of syringes to get the target amount out with that interval. So we would use the amount of syringes that we counted out. We would, and then after drawing out half of the vial, you would think, or what you're supposed to do is you're supposed to have 5 left. If it's a 10 ml vial, you're supposed to draw out 5 and have 5 left. But because of hub loss, there's less than 5. So what we did is we measured how much was left after half of the volume was drawn out. And that was, we used that to calculate our hub loss. The results from these tests showed that our hypothesis was fairly correct in that the number of syringes did increase the hub loss of each vial. But there was very inconsistent results in the raw data. And we think most of this is because of inaccurate starting volumes with our vials. So we were supposed to have 10 or 20 ml vials to start with, and we were depending on that assumption to make our calculations for hub loss. But because we were using a 30 ml syringe to draw up 10 or 20 mils, we really didn't get, we were not sure if there was 22 mils in the vials or 11. So we're not sure if the inaccurate starting volume might cause inconsistency. Also, interpersonal variability, as we saw before, does make things a little bit inconsistent. And unstable air pressure within the vials, as we're withdrawing liquid constantly from these vials, there's pressure building up and trying to pull the plunger back as you're making your measurements, which might also cause inconsistency. One of the things that, other than finding the range of hub loss, one of the things that we saw here is that we can improve some techniques within the lab. So the reason why we use 30 ml syringes to fill up these vials was because that is often how drug dilutions are made in the lab. So what we're thinking about doing, because of our inaccurate starting volumes, it would probably be more precise and better for our lab to start using smaller syringes and needles to mix dilutions. We'd also like to switch to a tuberculin syringe for certain procedures that do not need the needle to be changed, so that less controlled substances are wasted. Another thing we would like to change is add a column to these drug logs that lists the number of syringes used for that vial, because variables like the interval of withdrawal or the number of animals being treated does not actually affect the hub loss. So one thing that we think it would be reasonable to allow the 2 standard deviations from the mean of the number of syringes used per vial in our data would be reasonable to make for an expected range of hub loss, because we actually did not go over 2 standard deviations of the mean in our data. We actually went under a couple of times, so it would be reasonable to apply that in drug logs. In future studies, we would like to have more accurate starting volumes by downsizing the syringe that is used to fill the empty vials. We would also like to weigh the vials before using them, so that way we know for sure there are 10 or 20 mils only in that vial. All 3 syringe types would be tested in part 2. So in part 2, we actually only tested the vivarium syringe instead of the other 2. So we would like to test all of them for more data and more understanding of the study. We would also like more people to participate, because 3 people is not a very good representation of the entire population. So we would like to have more people test the syringes. We would also, one thing we ran out of time for, we were originally planning on reviewing the past drug logs of the mouse biology program to see if they were within that reasonable range, but we ran out of time. So we'd also like to do that later. And another thing is, because in a lot of procedures, you need to change out the needle. There's such a thing called a filled syringe, where it's just like a conventional syringe, so it has an interchangeable needle. But instead of a normal plunger, there's an attachment, an extension to it, that goes into the hub and gets rid of that liquid. So we would like to investigate that syringe a little bit more and think about switching to that as well. I'd like to thank Dr. Kent Lloyd, the mouse biology program, Dr. Kristen Grimsrud, Leslie Stewart, and Zoe Shee for making this possible. Any questions? Yes.
So you mentioned earlier that you did both a half contents test and an entire contents. I was wondering why you couldn't talk about the entire contents test.
So the problem with the entire contents test was that in the half contents test, you could see that there was a set number of syringes used for each vial. But in the entire contents test, we would lay out the number of syringes that would hypothetically need to be used. So if we're drawing out 10 mils with 0.1 intervals, with 0.1 intervals, you would think that you would need 100 syringes doing the math. But because of hub loss, you actually can't use 100 syringes to pull that out. So instead of 100, we'd lay out 100, but instead of 100, we'd only use 42. Or the next vial, we'd use 30. Or the next vial, we'd use 60. It was a different number each time. So it's hard to graph that in the raw data because of the inaccurate starting volumes. Whenever I tried to graph the raw data, it actually had a negative slope because the starting volume probably was not accurate. So the more syringes that were used, there was less, but there was either less or more volume to start with. So the more syringes used actually didn't represent hub loss. It just represented the amount that was originally in that vial. So we couldn't include it because it didn't represent hub loss. But the averages of each one, whenever you'd only take the averages and not the raw data, the slope was going up and showing that the number of syringes did increase hub loss. Yes?
Did you, two quick questions. Did you only use water in your measurements?
We only use sterile water. And another thing that I didn't mention was in the mass test, the reason why we used water is because 1 gram of water equals 1 ml of water. So it's really easy to translate between the visual and the mass test.
Thank you, Allie.
So next I'd like to introduce my colleague, Darren Tom.
Hi there. So for those of you who don't know, my name is Darren Tong, and I work in the mouse biology program of UC Davis underneath the mentorship of Kayla, Diana, and Dr. Wood. The mouse biology program is an organization that focuses on doing research into mice, as well as developing transgenic mice and selling them to researchers. Before we get started, I want to give you guys a little bit of an introduction as to what transgenic mice are. Transgenic mice are essentially mice with artificially induced mutated genes. This is created through a complicated multi-step process which I shall be guiding you guys through. Up here, we start with a CRISPR-Cas9 tool. A Cas9 protein reads the CRISPR RNA sequence and it makes edits to another RNA. This modified RNA is then injected into a fertile egg through a process called pronuclear injection. This fertile egg is then implanted with a surrogate mother mouse, and if all goes well, a mutant pup is born. Transgenic mice are used for a variety of reasons, such as science and commercial reasons. Commercially, as I mentioned, organizations such as the MVP create and sell transgenic mice. Scientifically, they can be induced with genetic diseases such as cancer and be used as a test bed for various treatments. One of the steps I mentioned earlier was pronuclear injection. And you may be wondering, what is pronuclear injection? This is the process in which something is injected into an embryo. It requires a high-powered microscope as well as 2 mechanically operated arms controlled by microinjectionists. One arm holds the hub over here, which holds the egg via a low-powered vacuum. The other arm holds the microinjection needle over there, and that injects the RNA. The microinjection needle is small enough and sharp enough to pierce the outside of the egg, but does not deal enough damage as to cause premature death. Because the amount we inject is so small, usually less than 2 microliters, we have to use a specialized machine, such as this one. In our experiment, we used a Narashiki IM300 microinjection. This is the machine we used. As great as transgenic mice are, they do have problems. First of all, they can be expensive. Individually, mice aren't that expensive, but when you have over 1,000 mice per year, the price really adds up. In addition, there is a low success rate. This is because the process is so complicated, so many things can go wrong, such as bad DNA design or not enough fertile eggs to inject. Here's a picture of a chimera mouse in which you can see it has traits of both a black mouse and a white mouse mutated together. So now you might be wondering, now what exactly am I doing about this? I'm working as part of a larger research project that aims to reduce the amount of mice needed per year. My part is to take a look at the number of eggs that are left behind after pronuclear injection because they are thought to not be fertile, and then from there, determine how many are actually fertile. I'll do this in 2 steps by first analyzing the eggs and then determining a solution. Hypothetically speaking, a large percentage of the fertile eggs will be lost. Our eggs were obtained from 5 female black mice through a terminal procedure in which the mice were killed by carbon dioxide asphyxiation. In addition, the mice had their oviducts removed. The eggs were removed from the oviduct and then stored in a liquid media. The media was submerged under mineral oil in order to prevent it from evaporating. Here's a picture of some mice eggs. They are currently in their 2-cell stage. All of these are stored in a petri dish and then from there in a carbon dioxide incubator such as this one. The carbon dioxide incubator was set to a temperature of 32 degrees Celsius as well as an ambient atmospheric concentration of 5% carbon dioxide. When we observed the eggs, we categorized them into 3 sections: unfertilized, deceased, and fertilized. Unfertilized, over here, had the appearance of a smooth circle along with a complete membrane on the outside. Occasionally, they will have a polar body, that small thing on the side. In addition, there are deceased eggs. Deceased eggs have a vastly different appearance. They are broken up into multiple chunks, and they tend to have no membrane on the outside. Fertilized eggs have an exceedingly similar appearance to that of an unfertilized egg before they enter the 2-cell stage. The main difference is that 1, they have 2 pronuclei, and 2, they have a higher chance of having a polar body. The 2 pronuclei can be identified by looking for the 2 circles in the center. They contain genetic information about the egg. During the experiment, over the course of 3 days, we took a look at over 600 eggs separated into 10 different batches. We observed them at 3 time points of each day: 11:30, 2 o'clock, and 4:30. We collected a massive table, and then from there, analyzed it and put in calculated averages, and then from there, percentages. Here are my results. As you can see on day 1, basically no embryos showed any signs of fertility. In addition, the percentage of eggs dead was at 33.90%. The rest of the eggs were unfertilized. On day 2, drastic changes began to happen. Around 32.82%, give or take, showed signs of fertility by being in the 2-cell stage and beyond. In addition, the percentage of eggs that were dead remained at a cool 38.23%, marginally larger than last time. On day 3, once again, there was a drastic change. The majority of eggs that are fertilized are now dead. Only 6.44% of the total remain fertilized. In addition, the average percentage of eggs dead was at a whopping 76.55%. It is likely that the remaining unfertilized and fertilized eggs will die within the next few hours or day. So the question is, what do we do about this? As you can see, there is a large percentage of unused embryos. The quickest solution to this is to have 2 shifts, a day shift and a night shift. The day shift will operate quickly after removal of the eggs from the oviduct, and then from there, remove any eggs that show signs of fertility. The night shift will wait around 12 hours, and then enter the lab and remove any eggs that now and later showed signs of fertility. However, this is both inefficient and wasteful, as it requires too many resources to create an entire separate set of staff, and in addition, is essentially a drain on organization resources. I propose 2 courses for future research. One will be to determine the optimal timing of pronuclear injection. By determining the optimal time, the most eggs can be found to show fertility, as well as the fewest eggs that are dead. If research is done, it will be if research is successful, it will help microinjectionists determine which eggs they should inject. This must be done both quickly and efficiently, however, as if it is too late, fertile eggs may die. My second solution is to determine which eggs are fertile without current observable traits. This is currently no such method exists, but if it does, it will be a complete game changer. By doing this, the microinjectionists will have no need to look at the classical traits of fertility, by which I mean the two pronuclei, as well as the polar body. In addition, it will allow microinjectionists to inject at any time immediately after removal from the oviduct, as they do not need to look for visual cues. If you were to ask me, priority should be placed on this, as if this is completed, there will be no need for the first solution. Here are my references. And before I finish, I want to thank a few people. First, I want to thank my roommate, Ben, for more support. I also want to thank the entirety of the mouse biology program, especially Josh, Diana, and Kayla. Thirdly, I want to thank Tyler. He is my counselor, and he's been excellent support. And finally, I want to thank all of you guys here at YSP for being good people and friends. Thank you. Before I end, are there any questions?
What exactly is Cas9? Is that a protein?
Cas9 is indeed a protein.
And what does it do?
It reads so CRISPR-Cas9 works by having a custom sequence injected, or put into an RNA sequence, right? The Cas9 protein reads this, and it uses it to edit other genes based off of the red sequence.
What's happening in between day 2 and day 3 that causes so many of the eggs to die?
Chances are the media is either getting old or the eggs are just getting old, and they don't have any life support from, say, a mother mouse, so they die.
Why was the error in the table a bit high?
So the main reason for that is that our batch sizes vary largely, mainly due to the oviducts and how many eggs they contain. For example, one of them contained 15, whereas another one contained over 100. So because of that, it's easy for the numbers to shift widely. Any other questions?
So you said you use CRISPR to, so what are you doing with CRISPR?
I'm mainly using it to edit RNA and then inject it into an egg.
Okay, why, is that a standard procedure for editing DNA?
Currently, yes, it is a relatively new technique. The protein and the idea itself was pretty old actually, but the idea of using it for gene editing is new. And we're using that because it's both efficient and cheap. Yes?
When injecting the embryo with the RNA, is there any lasting scars that you see in the developed animal from this injection?
Occasionally, it may result in death of the egg. Usually, though, microinjectionists are highly trained, and it's unlikely for this to happen. Anything else?
So you said you had a batch size of over 100. How many eggs did you analyze?
Over 600 eggs.
Well,
This is separated into about 10 drops, I believe, and each drop counted as a single batch. Yes?
Okay, sorry. If you're looking at so many eggs, isn't there a significant time difference between the first egg you looked at and the last egg you looked at?
Right. So we actually separated it into multiple days. So on day 1, we only looked at 4. And then on day 2, we did it in 3 different sessions, basically.
Okay.
And then after 3 days, we moved on to the next set. And then after that, we moved on to another set. Any other questions?
Thank you.
Next up is my colleague, Ellie.
Ellie, we're gonna wait just a couple minutes. We're 3 minutes ahead of schedule. Okay.
you can go get ready.
Then we're gonna go ahead and get started. We're back on time. So whenever you're ready, Ellie.
Okay. So, good morning everybody. My name is Elis Stoyanou, and for the past 6 weeks I've been doing research in the Special Cognition Laboratory in the Department of Psychology at UC Davis. Under the mentorship of Mike Starrett, I've been investigating simulator sickness during an immersive virtual reality application. So before we even start with simulator sickness, what is virtual reality? Virtual reality is a three-dimensional, interactive, real-time, realistic, computer-generated simulation that provides direct input to our senses. Virtual reality is used across a range of divergent areas, ranging from military and aerospace education to research and commercial uses. With the recent advancement of virtual reality, and especially of the immersion that the users feel in it, a new illness has come to light called simulator sickness. Simulator sickness is described as the discomfort that the users feel during and sometimes after a session in a simulated environment, and is often compared to the way we feel when we read a book during a car ride, when we get seasick, or generally when we get motion sickness. In fact, back in 1993, Kennedy and his colleagues showed that simulator sickness is a subform of motion sickness. Thus far, research has shown that these are the basic characteristics and similarities of the two distinctive conditions. So whereas in simulator sickness, the key point is the visual stress that includes symptoms like eye strain, headache, and dizziness of the head and fullness of the head, in motion sickness, the key point is gastrointestinal distress, which includes symptoms like nausea, burping, and stomach awareness. While simulator sickness is less severe and does not require true motion, motion sickness occurs when the sensory input provided by the eyes, the vestibular system, and other sensory receptors is at variance with what the central nervous system expects to receive. Before I move on and explain the cue conflict theory, which is believed to be a shared cause between these two, let me take a few minutes and explain the basic characteristics of the vestibular system. So the vestibular system consists of the semicircular canals and the otolith organs located somewhere inside there. The semicircular canals provide information about the angular velocity, and the otolith organs about the linear acceleration and the gravity acting on the head. So the vestibular system in total is responsible for balance and postural equilibrium. So what happens, what the cue conflict theory says, is that under natural conditions of self-propelled locomotion, all the sensory receptors of the basic orienting system transmit information correlated with each other. In a wide variety of situations, however, the harmony that exists between these functionally related receptors can be disrupted, and as a result, the combined influx is incompatible with what distorted expectations. Another possible cause is the inaccuracy of simulating motion in a simulated environment, which is believed also to go back to the cue conflict theory. So, can we reduce simulator sickness? Simulator sickness is polysymptomatic and polygenic, meaning that it includes a lot of symptoms and thus varies and depends on a lot of variables. So measuring simulator sickness has been a hard thing to do. The Simulator Sickness Questionnaire was developed in 1993 and is the most commonly used method of quantifying simulator sickness. As I've mentioned, virtual reality is used for military and aerospace training, so we have a lot of insight about how to prevent simulator sickness conducted by Army Research Institutes, and earlier this year, Fernandez and Foyner found reducing simulator sickness when restricting the field of view of the user inside it. So, I mentioned that I've been doing my research in the psychology department, but yet I'm studying simulator sickness. What is the connection? So, the immersion that the users feel, the immersion that the advancement of the virtual reality feel, allows the subjects of the research, like of a research in a spatial cognition laboratory like the one I'm doing research in, to get proprioceptive information. Proprioception is essentially that I know where my hand is right now, and I don't have to stare at my feet to know where they are exactly where I'm moving. So in a research conducted in 2006, showed that proprioceptive information provides significant difference in the research site that they actually developed. So this is why my mentor has been using an immersive virtual reality while studying the spatial memory of his subjects. Mike Starrett is researching the effects of vestibular, head-based, and proprioceptive, body-based information in our spatial memory, as well as how the geometry of the environment influences our spatial representations by having his subject navigate in a virtual city. the immersion in the virtual environment is accomplished with the commonly used head-mounted display and the omnidirectional treadmill. Omnidirectional meaning that the subjects can rotate 360 degrees without actually leaving the room and being able to provide the proper set information. So this is what a subject looks like during a training session. All subjects have to undergo this training session in order to be smoothly accumulated in the simulated environment. And what the experiment is basically about, the subjects navigate through this virtual city where 8 special targets are sparsely located inside it, and then they're tested on their spatial memory. The problem is that 30.2% of the subjects had to withdraw due to simulator sickness. So what I've been testing is what makes someone susceptible to simulator sickness, as well as is there a way to predict and possibly prevent a simulator sickness experience. So in addition to already collected data about their spatial memory, I could work with these information collected from the subjects, such as age, stress, and the last 27 subjects of my research completed the simulator sickness questionnaire once before the study began, and once they have completed the experiments, or they had to withdraw because of simulator sickness. So what we did with this data is that we compared the group averages of the ones that withdrew from the subject study and the ones that completed the study across a number of simulator sickness related factors, such as sleep, for example. We did that through the student t-test, where we tested the difference of the means divided by a measured variable of the 2 samples. And when we had the results that showed significant difference, so we had the factors that could work as predictors, we plot them in a logistic regression. We chose the logistic regression technique because the outcome is binary, right? So it's either you get simulator sick, either you don't get simulator sick. So we wanted to test the probability of getting simulator sick across a number of different predictors or independent variables such as factors. So this is what my t-test looked like, some of it. I have 2 examples. The first one showed significant difference. So the p-value in this one is lower than 0.01, that's the stars there. But this one, as you can see, the difference between the means of the hours the subjects slept is not significantly enough different. So the 2 factors that showed significant difference were experience with video games, something that the subjects self-rated themselves, and treadmill proficiency. Treadmill proficiency was a subjective measure that the experimenter took a note of during the training session, so it was the first thing, and it was concerning the subject's capability of walking on the treadmill, the first thing he took a note of during the study. So these are the regressions. So what we can see in that, the first thing that I would like to highlight is that we had data points for all this. So we had subjects with gaming experience rated at 1, which was low, that completed the study. And we had subjects being able to walk on the treadmill very well that had to withdraw from simulator sickness. The assay term, the sigmoid, showed that the probabilities significantly changed at the inflection point where there is 50-50% of getting simulator sick and withdrawing from the subject or the otherwise. The left, upper high left quadrant shows where the probability is higher. This is the graph I showed earlier, right? This is the linear function that we use to predict the data, and with every change in the x-axis in the predictor, the log of odds changed by the slope of the line. So through a simple transformation, this allows us to compute the odds, and through another simple transformation, this allows us to calculate the probability of a subject withdrawing based on these 2 factors. So what the Spatial Cognition Laboratory might consider of actually doing is of creating another inclusion criteria such as gaming experience, because we don't want our subjects to withdraw due to simulator sickness, right? So if a subject has gaming experience lower than 2, they might consider saying that we have this problem with simulator sickness, so please take responsibility of that on yourself. Also about the treadmill proficiency, what could be done is they could add another training session, so the walking abilities of the subject on the treadmill enhance before they actually get simulator sick. Further research might consider having a larger sample to investigate simulator sickness, and thus being able to explore whether individual factors such as how much, when they slept, how much they slept last night, or when they ate, could influence simulator sickness. If I were able to collect my own data, I would find it very interesting to test whether there's a significant correlation between the chronological age of the subject and the simulator sickness, because research shows that there is relationship between motion sickness and chronological age. So children falling under the age of 5 years old are more susceptible than we are. We seem to be able to adapt to the motion that we feel. Also, further research might explore whether the inappropriate usage of the treadmill could lead to the cue conflict theory and thus simulator sickness. So at this point, I would like to thank Mike, my mentor, for all his guidance and his help, Dr. Arne, his supervisor, the Spatial Recognition Laboratory in Tolo, Dr. Pomeroy, the counselors that for all the help they offered, and everybody. So these are my references. Any questions? Yes?
So could you give like a specific example of cue conflict theory in a simulated environment?
So what happens is that you're in a simulation and your visual information says that you're moving, but your vestibular information says that you're in a stationary position. Like that. Yes.
For the graph of the odds, what does the blue line mean and how do you use that in your graphs?
So, interesting to mention is that the inflection point, where the probability is at 50%, is the inflection point exactly where the odds are 1 to 1, meaning that these two graphs show exactly the same thing. At 2, the inflection point higher rises, so the probability as well rises as well. I hope I explained that. Yes.
In general, did participants tend to withdraw earlier or later during simulation?
So what happened is that when they express discomfort, they were asked to exit the virtual environment by taking off the head-mounted display, and after like a short break, they were asked, would you like to continue the study or would you like to abort? They tend to continue, but if they did show again simulator sickness symptoms, they were asked from the experiment to withdraw. Yes?
Were participants given a path to walk throughout the virtual reality, or were you allowed to free roam?
So in the virtual city, you're free to navigate wherever you want, but you cannot pass through the walls of the buildings. So yes, like walking outside, I guess.
One of the things I learned is that our first draft requires over 80 frames per second in order to prevent nausea. Did this play any role in your experiments?
Any extreme conditions like emesis? So like,
for nausea, it's lower than frames per second.
so nausea is not one of the basic symptoms in simulator sickness, while it is one in motion sickness. Only 2 of the subjects had problems with extreme conditions as emesis. Yes, Ben.
So, it looks like it's before the inflection point, the data looks quite linear, but after the inflection point, it looks logarithmic. Do we know why that is, or am I just seeing?
So the R-squared for the probability graphs is approximately 13 to 16%, which means that the R-squared shows how well our model fits our data. Maybe if we had a higher sample, like larger sample, we could get the S-curve, like the sigmoid curve, better shaped, but at this point, we get that it drops significantly here, and it looks like it raises the probability in the left quadrant. Yes.
Okay, so you said that the treadmill, like how well people use treadmill was rated by somebody else, but the gaming experience was self-rated. Do you think that played any factor, or do you think people were
So
honest?
even though the treadmill proficiency was a subjective measure completed by the experimenter, it was standardized for all of the subjects. The gaming experience, we trust our subjects. Maybe yes, but we hope that they were sincere. Yes, Renee?
How long was the average session for the Rachel session?
It was, is there another?
That's
Okay, so it was like, the training session, it was like 20 minutes, and the actual experiment was like 1 hour and a half to 2 hours.
And did subjects like, do another day of that session, or did they only do it one time?
One time. Anything else?
Last question.
Could having the same subject perform the session through multiple times in a week affect their experience with the sickness?
Oh, so if we, maybe having the subjects have the training session a day before or another day, separate day from the experiment could help, but this was not done. But interesting question, maybe can it be examined further research?
Thank you.
So up next is Gregory Martin, my colleague and my friend.
Hello everyone, my name is Gregory Martin, and I worked in the Franz lab where we were studying the growth of an algae species named Chlora soriciniana in wastewater for the production of biofuel. So what is algae biofuel? Algae biofuel is a biofuel made from the lipids, which are like the fats and oils from algae. It's one of the most promising sources of biofuel that we currently know of. As you can see in this picture, it requires the least amount of land compared to any other biofuel crop, which is good because we are running out of land. And it produces a carbon neutral cycle. This is because when you make a renewable fuel from algae, the CO2 that is burned is absorbed when the algae is grown. So no actual CO2 is produced, although the car would still emit emissions. It is accounted for when the algae is grown. The only problem with algae biofuel is that the costs are still too high. It costs around $7 a gallon to produce a gallon of biofuel. This is of course way more than crude, so that price needs to be brought down through trials and experiments to reduce the growing costs and the processing costs. Specifically, I looked at the growing costs, and I took an angle to reduce the cost of the media. You have two options to grow algae, either to grow it in some sort of wastewater or to grow it in a laboratory-made synthetic media. We chose to use wastewater, which is a very cheap source of a nutritious media, to grow our algae. Wastewater is great because it is cheap, as I said. It's renewable, and it is green because the algae actually clean the wastewater up as they grow. One of the wastewaters that I worked with was dairy wastewater. Dairy wastewater is basically untreated liquid manure and runoff from a dairy farm that we stereofiltered to 0.22 microns. The other wastewater that we worked with is anaerobic digester effluent. This actually came from the on-campus Reed facility, which is this picture. Basically what happens is that you chop up a bunch of organic waste and you put it in one of these big tanks with bacteria and you seal it up, and the bacteria digest the anaerobic waste, or digest the waste anaerobically and produce methane, which is used as a biofuel, and also effluent, which is a nutrient-rich liquid that is full of all of the not all the way digested materials. Once this effluent is put through ultrafiltration, which removes any solid particles, it is then called permeate, and then we take the permeate and strip it of most of its nitrogen, so it's down to about 5 to 10% of its original nitrogen, and then that's what we use as our anaerobic digester media. So this is how I grew my algae. It was grown in 6-well falcon plates on a plate shaker under artificial light. Each trial was run in n=5, so I had 5 different wells of my algae growing, along with 1 blank to use as subtraction. We also grew a synthetic media plate as a control to see how that growth would compare to wastewater. For testing, We took an initial photograph of every plate, just with a camera. Then we took the absorbance at 750 nanometers. Absorbance is basically measuring the optical density of the algae. So you shine a light through, and then you measure how much of the light comes out the other side. Chlorophyll fluorescence, which measures the fluorescence of chlorophyll. And we did microscopy at 60x, which looks at the morphology and gives an idea of cell density and cell death. So this was my first experiment, where we looked at the growth of Chlorella sorokiniana in dairy wastewater. We took 5 different concentrations, 100%, 75%, 50%, 25%, and 10%, and grew these plates for 10 days. As you can see, so most of the cultures grew fairly well, growing about as well as the synthetic did. And those were the 10%, 25, and 50%. But anything over 50% dairy wastewater resulted in very little growth, and the 100% had no growth. These are microscopy images from that trial. They all started out fairly similarly, but by day 7 you can see that 75 had very little, and the 100% also pictured here was completely dead. This is a similar experiment, but done in anaerobic digester effluent. We used 100%, 90%, 80%, 70%, and 50%. And with the anaerobic digester effluent, the algae grew well in almost all the concentrations of it, and really showed no detrimental effects at the higher concentrations when compared to dairy wastewater. Again, here are some pictures from that trial. As you can see, all of the plates show definite signs of growth. So my next experiments, based off of those experiments, the first one that we tried was we looked at the differences between 50 and 75% dairy wastewater. This is because at 50% they showed very large amounts of growth, and at 75 it was very small. I wanted to see if we could find the exact point where there was too high of a concentration of dairy wastewater for the algae to grow robustly. Next, we mixed the ADE, which has had most of the nitrogen taken out of it, with permeate, which has the full amount of nitrogen, to see how much additional nitrogen and ammonia the algae could handle. And finally, we spiked the 100% nitrogen stripped ADE with ammonia, again to see if we could get an idea about how much extra ammonium alone the algae could handle. So this is the first experiment, which was in dairy wastewater, where we looked at a refined concentration scale. And as you can see, all the data graphed out very nicely. So this is the 50%, which again showed very similar growth to the synthetic culture, followed very closely by the 56% dairy wastewater. But anything over 56 started to show detrimental signs of growth. And then the 75, again, did not grow very well. So from this graph, I would say that anything over 56% really starts to show detrimental signs of growth. And then this is a picture of 2 plates. So as you can see, the cells here in 50% are very dense and green and happy looking. And these cells are very sad, and they're very pale. So this is the ratios of AD and permeate. This experiment was to see how much extra nitrogen and ammonium it could take. So this is 100% ADE, and then this is 95-5 ratio, so 95% ADE and 5% permeate, which still grew fairly comparably to 100% ADE, but anything over that just killed the plate, as you can see by all the lines stacked here. This is comparing the 95-5 and the 90-10. So there was very little growth here, versus there was quite robust growth there. The final experiment looked at spiking ADE with ammonia. We had concentrations of 0 millimolar, 1 millimolar, 1.5 millimolar, and 10 millimolar, and 3 millimolar. So 1 millimolar showed absolutely no effect on growth, but anything over that started to show detrimental signs of growth, whereas the 1.5 and 3 millimolar are there, and then 10 millimolar completely killed the plant. This is comparing the 1.5 millimolar, which grew fairly well, with the 3 millimolar, which didn't grow so well. As you can see here, it's kind of hard to see, but all the cells are very sort of gray and not green, dead. And then here they're all green and happy. So this is as a summary. Growth in low concentrations of wastewater, around 50% dairy wastewater and 90% ADE, is very comparable with that growth in synthetic media. This is very promising for dairy wastewater as a growth media in future industrial and research applications. The maximum concentrations that I would recommend for growth in dairy wastewater and anaerobic digester effluent are 75% and 95% respectively. Anything over that, and there are large amounts of cell death, and it just doesn't work out well. Optimum concentrations for growth are much lower though, around 10% dairy wastewater and 50% ADE. That's probably due to the amount of inorganic growth inhibitors, organic growth inhibitors that are in the media that stop the algae from growing well at high concentrations. So future research needs to be done in characterizing the lipid production and biomass production of these cultures to see accurately how they would perform in a biofuel situation or like a feedstock situation, and what exactly would be their efficiency. Another future research could be on other wastewater sources like municipal wastewater, or piggery wastewater, or brewery wastewater, where you could see if chloros or pignata might grow better on those wastewaters compared to dairy wastewater and ADE. Another interesting study would be looking at longer growth periods. The place that we had, even place that didn't grow in the beginning, if we left them for long periods of time, they would eventually start growing. And this is hypothesized to be due to the volatilization of ammonia. But we're not sure, and if we did some testing on that, we could figure out what's causing that. And finally, we could do testing on artificial selection to see if over time we could grow out strains of algae that were immune to high concentrations of wastewater, so that we could use larger concentrations in our lab. So I'd like to give thanks to the France group for letting me invade their lab and teaching me so much. And to YSP for giving me this great opportunity, and to the National Science Foundation and California Energy Commission for providing grant money for the research. Okay, any questions?
So you're using algae to create biofuel. Can that biofuel be used in modern day equipment such as cars?
Yes. So there's a process called transesterification, which basically, as you have like a triglyceride, it has the glyceride backbone and then it has 3 hydrocarbon chains. And transesterification just takes that glycerol off and then turns it into some long chain hydrocarbon fuel that can be used in most any diesel car without modification.
Your lab decided to work with Chlorella sorghiniana, but do you think there are other algae species that would be interesting to look at?
Definitely there are other algae species. There's like there was a large like effort a couple of years back to look and find exactly what species create the highest amount of lipid and lipid productivity. But not that much research has been done to like find the best wastewater growing species. So our lab knows that Chlorella sorghiniana grows very well because of its own experience, but there are some other species like Phaeodactylum tricornutum, which works really well, and there is also Nanocoapsis sp that works well, and there's some other species floating around too. Sunny?
You said you stripped the nitrogen before further processing. Why did you do that?
So that was because the nitrogen, and especially ammonia, which is considered a nitrogen source for the algae, is a growth inhibitor. So if there's too much of it in the media, the algae just won't grow, which is sad.
So do you know what specifically is causing the different amount of clinical rates between the two wastewaters and why it becomes toxic at the higher level?
So that would be hypothesized to be due to the fact that the ADE is digested back by bacteria. So a lot of the more complex compounds have been broken down to simpler compounds versus the dairy wastewater has gone through no digestion whatsoever. So there are probably some larger organic or inorganic compounds that are in the dairy wastewater that are harmful to the algae's growth that in the ADE are broken down.
I think in your first graph that you're growing, the first one, the highest concentration, the line for the graph first went above the level where it plateaued out and it went back down. Do you think could be a reason for that?
Algae cells, when they get stressed out, which can happen from either having like too little amounts of concentrations in their media or too much, will actually like enlarge themselves because they'll make more lipid. So my guess would be that the starting population survived for a little bit in that media, enlarged itself because it got stressed, and then immediately died off, which would cause it to go up a little bit, plateau, and die. Because they weren't actually growing and dividing, they were just sort of living and dying.
All right, we'll be taking
Okay, thank you.
Don't worry about it.
I don't think anyone
All right, let's settle so Logan can get started.
Hi, my name is Logan Samuels, and I work in the Department of Neurobiology, Physiology, and Behavior under the mentorship of Dr. Aldrin Gomes. And what we looked at is the effects of indomethacin on cardiac protein degradation in mice. So NSAIDs are non-steroidal anti-inflammatory drugs. And one of the biggest NSAID is ibuprofen, which is over the counter. So these are the most commonly used drugs in the world. And in this graph done by the American Nutrition Association, we see that NSAIDs account for over 16,000 deaths each year. And this graph was done in 1999, and the popularity of NSAIDs are increasing each year, and today this number would be much more. So indomethacin is a prescription NSAID, and is commonly prescribed for inflammation, pain, and tendinitis. And it was found that peak plasma concentrations were 1 to 2 milligrams per microliter in the blood, which was done in a previous study. And we calculated this to be 7.9 micromolar. So protein degradation is done through the 26S proteasome and the ubiquitin structure enzyme. So the 26S proteasome is composed of two parts. The 19S proteasome, which is the two caps on the top. And what this does is it finds the proteins that need to be degraded, and then it denatures them and transports it to the 20S proteasome, which then degrades the protein. And then you have ubiquitin, which tags the actual proteins that need to be degraded so that the proteasome could come and find the protein. So the way that this works, which is known as the ubiquitin proteasome system, is you have an E1 enzyme which tags onto the ubiquitin with the use of ATP. And then this E1 enzyme takes it over to the E2 and E3 enzymes, which are interacting with each other. And what this E2 and E3 complex does is it brings it over to the substrate, which is the protein that needs to be degraded. And it keeps repeating this until a polyubiquitin chain is created in order for the 19S proteasome to come over, degrade it, and transfer it to the 20S proteasome in order to get rid of the target protein. So NSAIDs have been linked to cardiac function, where they cause dysfunction. So in a study done by Dr. Aldrin Gomes, he found that two NSAIDs, methylphenamate sodium and naproxen, were found to have increased levels of ROS, which are reactive oxygen species. So what this ROS does is it causes a buildup of these oxidized proteins, which is bad for the heart. But what he also found was that in MS, there was a decrease in beta-5 activity, and this results in less 20S digestion from the proteasome, which, when this doesn't work, the heart does not work as well. And for the reactive oxygen species, he found this through an oxyblot, which I'll explain later. And he found that there was more oxidized protein in the methylphenamate sodium. So the way that I tested the effects of indomethacin on cardiac protein degradation is I received heart samples that were done with 4 control mice and 4 injected mice with indomethacin. And these mice were exposed to 1 milligram per kilogram of indomethacin a day for 5 days. And this is considered a low dosage of indomethacin over a lengthened period of time. So to prepare the samples, I took the hearts and homogenized them by crushing them with liquid nitrogen and dissolving them in urea. And then I used a nanodrop machine to find the concentration of protein in these samples. And based on these readings, I diluted it to 0.6 milligram per milliliter. And in order to run a gel, I then took part of this solution and put mercaptoethanol to break the disulfide bonds between the proteins. And we did this because if proteins are bonded to each other, they cannot be denatured. So once we broke these disulfide bonds, we heated the samples to 96 degrees for 5 minutes in order to denature this protein so that it could be ran through gel electrophoresis. And this is an example of the concentration test I did. And you can see that it starts at a higher protein size and goes lower. And we found that the bands were very similar, so these samples were ready for experimentation. So the way we experimented was through the Western blot, which involves gel electrophoresis, as I just explained. Then we transfer this to a nitrocellulose membrane. And what this does is it takes the proteins from the gel and puts them onto a membrane so that we can perform certain tests on it. So after we do this transfer, which you can see in this image, and we put it into a transblot machine, which does this in 7 minutes, which is fairly quickly, we then image it using Ponzo dye in order to get an image of the bands. So after we destain, we block it with non-fat milk. And we do this because the nitrocellulose membrane is very sensitive. So it could have extra proteins that got on it besides the specific protein we're looking at. So in order for the primary antibody, which we use to link onto the target protein, the primary antibody will not link to random proteins, so we will not get as many artifacts on the images. So after we incubate it with the primary antibody, which latches onto the specific protein, we wash this off and then incubate it with a secondary antibody, which links onto the primary antibody. And what this does is it fluoresces so you can image it after using chemoluminescence. So this is an example of a protein Western blot that I did. And you can see that it starts at a higher level of size and goes down just like the gel. And I did this with control and indomethacin samples. So I used this exact blot in order to do my experimentation. So the first thing we did was test for PMSA6 expression, which is linked to proteasome quantities. And by incubating it with this antibody, we found that the control and indomethacin had similar proteasome quantities. And this is not a surprise because this was found with naproxen in previous studies done by Dr. Aldrin Gomes. So then we performed an oxidized protein expression blot. And what we found with this was very interesting. Although there were similar levels of oxidized proteins, which are linked to ROS, we found that after measuring the amount of DMPH in the oxidized protein, which is a residue found on the ROS, we found that there is a dynamic change within the levels of oxidized protein. You can see that in the second lane, there's a little more oxidized protein at lower levels, lower sizes, and that in another indomethacin sample, there's higher levels of bigger oxidized proteins, which we look to do future testing on in the future. So the next thing is we tested for polyubiquitin, which is the ubiquitin strands that are linked to the protein for the 19S proteasome to come find it and be digested. So what we found that there was a higher amount of polyubiquitin in the indomethacin sample, and this is important when analyzing our data in order to see what the indomethacin was actually doing to the protein. So next we tested for beta-5 activity, which is chymotrypsin-like activity, which digests proteins. So this is going on in the proteasome, and we found that the control and indomethacin had similar proteasome activities for the beta-5. And then we tested for beta-2, and beta-2 is trypsin-like activity, another form of digestion for proteasomes, and we found that again there was similar proteasome activity. So we can use this data to hypothesize two things that are theoretical that the indomethacin is doing to the cardiomyocytes. So the first thing that we can conclude is that there's an inhibition of 19S regulatory activity. And this makes sense because if we have the same proteasome quantities at the same levels and more polyubiquitin, this means that the proteasome is not targeting the polyubiquitin, and this polyubiquitin is stranded there and can't be degraded. So that means that the 19S regulatory proteasome is not doing its job, as it is not finding these proteins, this polyubiquitin, so these activities are being affected, which leads to the increase in UPS dysfunction and the increase in polyubiquitin levels. And another possible thing that is being affected is the E1, E2, and E3 enzymes that are part of the UPS system. And what is going on here is that this dysfunction is causing for slightly different amounts of ubiquitin to be latched onto the protein, which is a problem and will increase this polyubiquitin level. So in the future, we hope to look at the dynamic change that was happening in the oxidized blot, as different mice were having different responses, so something is being affected by the indomethacin. We also hope to do more biological assays to look at the 19S activity instead of the 20S, so we can see what exactly is going on and if this hypothesis is true. So in the future, this data can be used in assessing whether indomethacin should be used in short amounts, and whether a doctor should prescribe this medicine to his patient, as there are inherent risks with taking indomethacin, and this data would be very useful in making that decision. So I'd like to thank Dr. Aldrin Gomes for allowing me to be in his lab and mentoring me. I'd like to thank all members of the Gomes lab, Dr. Rick Pomeroy, Ann Ha, my group counselor, and all participants and friends I've made at YSP. So any questions? Thank you.
Hey, don't lysosomes account for a lot of protein degradation?
Well, that's actually a misconception. Proteasomes actually account for 60 to 80% of protein degradation in eukaryotic cells, and lysosomes are only about 20%. So they're teaching this wrong in high schools.
What are NSAIDs again?
Say again?
NSAIDs?
NSAIDs are non-steroidal anti-inflammatory drugs. So how they work is they inhibit cyclooxygenase enzyme activity. What these cyclooxygenase enzymes do is they inhibit the production of prostaglandin. What this prostaglandin does is it causes pain, and that's the sensation you feel if you get hurt. So NSAIDs block that pain. Yes?
So then is there pain blocking mechanism related to why they could conceivably be carcinogen?
Well, that could be a factor in that, but the pain blocking is, what we were looking at is the protein degradation and the effects it has on the cardiac system.
So with the protein, why do NSAIDs have protein degradation, or cause protein degradation?
Alright, so like I was showing you, it causes changes in proteasome activity. So if there's less proteasome activity, less proteins that need to be degraded aren't being degraded. So this causes a lot of dysfunction within cells and can lead to apoptosis.
Is this degradation part of the pain blocking response? Or the pain blocking mechanism?
No, it's not.
Okay.
Up next is my colleague and friend, Rebecca Chang.
Rebecca, is everyone who's here to see you present here?
Yes.
Okay.
As long as you're here.
Hi, my name is Rebecca Chang, and I have been studying the effects of restrictive cardiomyopathy, R145W mutation, on signaling pathways and proteasome function at the UC Davis Muscle Physiology and Proteomics Laboratory under the guidance of my mentor, Dr. Alvin Gomez. So restrictive cardiomyopathy is a rare myocardial disease that represents about 5% of all heart muscle disease cases. The average age of diagnosis is around 5 to 6 years old, and the disease is characterized by stiff ventricle walls where the heart has difficulty relaxing after blood returns from the body. However, unlike other cardiomyopathies where the heart is dilated or enlarged, you can see that restrictive cardiomyopathy hearts are generally normally sized or only slightly enlarged. Restrictive cardiomyopathy can also have genetic causes, which are sarcomere protein gene mutations, or more specifically mutations on troponin I, troponin T, or alpha cardiac actin genes. And you can see in the green part of the diagram that is troponin I, which is where the R145W mutation is located. This disorder is also inherited in an autosomal dominant pattern, which means that it only takes one copy of the mutant gene for someone to develop the disorder. So previous research relating troponin I mutations in restrictive cardiomyopathy have shown that there is an inability to properly inhibit ATPase and muscle force development, as well as having a greater muscle contraction sensitivity to calcium 2+ ions, leading to severe diastolic dysfunction. And this just means that the heart has difficulty relaxing after blood returns from the body. Currently, we are the only lab with the R145W restrictive cardiomyopathy mutation in a transgenic mouse, and the only other restrictive cardiomyopathy mouse model also showed impaired function in the heart. And so in my project, I worked with R145W and wild-type mouse hearts. And so first I powdered these using a tissue pulverizer, mortar, and pestle, then homogenized them in urea with a tissueizer, centrifuged them, put them through quantification, trypsin protein digestion, TMT mass tagging, mass spectrometry, pathway analysis, and finally a proteasome assay. So this is an image of a frozen mouse heart, and this is the powdered heart. And so during trypsin digestion, trypsin was added to the samples and it cleaved the protein, and during TMT mass tagging, the 6 regions, which are in those 6 bulk tubes, then tagged the sites where the protein was cleaved by the trypsin. So there are 6 different regions that all have the same chemical structure, but they differ in their isotopes, which are found on the mass reporter sections and the mass normalizer. And so they all have the same NH2 reactive group, but their mass reporters range in mass from 126 daltons to 131 daltons. And they're balanced by the mass normalizer so that the total mass of the region is the same for all 6 of them. However, during mass spectrometry, the mass reporter is then cleaved off, and because of their different mass, they're able to be differentiated on the mass spectrum. And so after mass spectrometry, we did statistical analysis to see which proteins were actually differentially expressed in the R145W hearts, meaning that they were expressed in either significantly higher or lower amounts than in the wild-type hearts. So one example of a protein that was differentially expressed was the proteasome subunit alpha type 1, and this protein was identified during mass spectrometry by these highlighted amino acid sequences. And the green parts are places where the protein was cleaved by trypsin and then tagged. And so looking specifically at the sequence in the red box, you can see that the sequence was read both forwards and backwards, which is what the red and blue colors represent. And so you can see they're on the spectrum. And then for quantifying these proteins, we can look at the cleaved off tags that are located in the red box on the spectrum. And so in the 6 regions, in 3 of them I added wild-type samples, and in the other 3 added R145W samples. And you can see that in the wild-type samples, there was more of the cleaved tags showed up on the spectrum than the tags that had R145W samples in them, suggesting that there was more of this proteasome subunit in the wild-type samples than the R145W samples. So proteasomes are a multi-subunit enzyme complex with inner alpha rings, inner beta rings, and outer alpha rings, and the subunit we just looked at is part of the alpha rings. And proteasomes are responsible for regulating proteins that control cell cycle and apoptosis, as well as protein degradation as part of the ubiquitin system. And because of their role, the inhibition of proteasomes can result in apoptosis and cell cycle arrest, but it also allows proteasome inhibitors to be used as a last-ditch cancer treatment because they cause cell death. And so after looking at which proteins were differentially expressed in the R145W hearts, we then looked at the pathways that these proteins were a part of. And the pathway that contained the highest number of differentially expressed proteins was the Parkinson's pathway. And although Parkinson's disease obviously cannot occur in heart cells, looking at this pathway can tell us the roles of the proteins that we found and how their downregulation may have affected the hearts. So in the Parkinson's pathway, we found these 3 proteasome subunits and these heat shock proteins were all downregulated. And because of the heat shock protein's role in responding to stress, we can hypothesize there is likely higher levels of oxidative stress in these hearts, as well as lower levels of proteasome activity due to the downregulation of the proteasome subunits. So this is an example of another heat shock protein that we found to be downregulated in the R145W hearts. And you can see again in the chart that there's a higher amount of the protein in the wild-type samples compared to the R145W samples. And this heat shock protein is part of the heat shock semi-chaperone family. And these proteins are basically responsible for responding to stress, response to unfolded proteins, and assembly of protein complexes, as well as transport of protein into mitochondrial outer membranes. And because of their role in stress response, that is why we hypothesize that downregulation of these proteins would lead to higher levels of oxidative stress. And this is an example of a heat shock protein. So after looking at signaling pathways, we also looked at biological processes that these differentially expressed proteins were a part of. And so we found that most of the differentially expressed proteins were part of cellular and metabolic processes, which is consistent with previous research that showed that higher amounts of ATP are needed per muscle contraction in restrictive cardiomyopathy hearts. And then knowing that the proteasome subunits were downregulated, we also hypothesized that there are lower levels of proteasome activity. So then we did a proteasome activity assay to see if this was true. And so in our assay, we prepared wells with sample that had with and without proteasome inhibitor, which would inhibit proteasome activity. And we also added a fluorescent substrate, which when cleaved fluoresces at a different wavelength. So then we put these wells in a fluoroscan, which detected the intensity of the cleaved substrate to tell us how much proteasome activity was in the wells. So looking at the chart, you can see that the wells with the wild-type sample indeed had higher levels of proteasome activity than in the R145W wells. And so in this study, we found that many biological processes were affected, mainly cellular and metabolic ones, which is consistent with previous research. And we also found that the signaling pathway with the highest number of differentially expressed proteins was the Parkinson's pathway, where both proteasome subunits and chaperones were found to be downregulated. So then we hypothesized that due to the downregulation of these proteins, there are higher levels of oxidative stress, as well as lower levels of proteasome activity, which we did find in our proteasome assay. However, we do need future tests to determine the actual levels of oxidative stress in these R145W hearts, and these can be tests determining antioxidant status of animals, the levels of ER stress-related proteins, and the reactive oxygen species formation in these hearts. So I'd like to thank my mentor, Dr. Alden Gomes, all the people from the Muscle Physiology and Proteomics Laboratory at UC Davis, and everyone from YSP for your support in this great opportunity. Thank you.
I'm not sure if I missed this, but why did you conduct the cleaving with trypsin earlier?
Okay, so we cleave the proteins with trypsin so that we can tag them. It's just for that.
Was there a reason for cleaving them at certain sites?
Not that I know of, but generally we use trypsin for protein degradation. Yeah.
Is there a perceivable advantage to having an R145W mutation, or is there just currently no other explanation for why it occurs?
What do you mean? Say your question.
Is there a perceivable advantage to having a mutated R145W in place of old something?
There's no advantage. This is a disease. Most people die after 5 years of diagnosis.
So you mentioned that the restricted cardiomyopathy is linked to Parkinson's because of the proteins involved?
So there's no Parkinson's. We just found that many of the proteins that were differentially expressed in restrictive cardiomyopathy were part of that pathway. So there was no Parkinson's in the heart, but they were part of the pathway, and we looked at the role in their pathway to see the effects that their downregulation would have on the heart.
And then just another question, do you think people with Parkinson's have a higher risk of developing restrictive
No, it's not related.
Okay.
Thank you. Sean, we're about 5 minutes ahead, so if you'll just hang tight for a couple minutes, we don't want to get too ahead, okay?
Okay. But next up is my colleague Sean Rowe.
Take 5 minutes. 5 minutes. No, we can wait. We'll wait a few minutes. I'll let you know.
One of my friends is going to come back to
Good, I've only got 2 left.
Now you can start.
Okay. Hello everyone, I'm Sean Rose from Spruce High School in San Diego, California. And for the past 6 weeks, I've been working under the guidance of Julian Helmine and under the supervision of Dr. Cecilia Giolivi in the Giolivi Lab in the Department of Molecular Biosciences. And we've been optimizing a PCR amplification strategy for detecting heteroplasmy in mitochondrial DNA with a mismatch-specific endonuclease. So mitochondrial DNA, aka mtDNA, is circular and consists of 16,569 base pairs. And it resembles bacterial DNA because one theory suggests that mitochondria were once prokaryotic cells that developed a symbiotic relationship with eukaryotic cells. So mitochondrial DNA is inherited maternally, and a few major structural important things about mitochondrial DNA is that it has no introns and very few non-coding regions. And mitochondrial DNA consists of 37 genes, 13 of which encode for protein subunits in the electron transport chain and are not encoded in the nuclear genome. So mitochondrial DNA is especially prone to mutations because its location is in the intermembrane space of the mitochondria and is exposed to free radicals. And it also has a limited capability in DNA repair enzymes and is unable to repair pyrimidine dimers. So in each mitochondrion, there are multiple copies of the mitochondrial DNA. And when you have the coexistence of different copies of mitochondrial DNA in the same mitochondrion, you end up with a phenomenon called heteroplasmy. And in normal healthy people, the levels of heteroplasmy are generally very low, but whenever the levels of heteroplasmy exceed the biochemical threshold, there can be detrimental effects to health. So in general, postmitotic cells have higher levels of heteroplasmy than other cells because they accumulate mutations over time. And high levels of heteroplasmy lead to different kinds of mitochondrial diseases. So for our study, we isolated mitochondrial DNA from genomic DNA with PCR amplification of these 9 fragments that you can see right here. And those fragments are overlapping and about 2,000 base pairs long. So once we have those PCR products, and if there is any heteroplasmy, when we denature and reanneal these 2 copies, we will end up with some heteroduplexes, in which one strand comes from one copy and the other strand comes from another copy. And there will be mismatches in the DNA where the base pairs do not follow Watson and Crick base pairing. And the severe nuclease, which is the mismatch-specific endonuclease, will find the location of these mismatches and cleave the DNA at that point, giving you 2 shorter fragments. And you can detect those shorter fragments by subjecting them to gel electrophoresis. So first, for a control experiment, we had 2 strands of DNA, a control G and a control C. And when we denatured and reannealed the control C, we would only get homoduplexes, so you don't get any shorter fragments cleaved by the nuclease. And when we hybridized the control C and the control G, we did end up with heteroduplexes that either had double C or a double G pairing. And as you can see, we got 1 band, although we should expect 2. Since these DNAs are 600 base pairs long, and the fragments should be expected to be 400 and 200 base pairs. The 200 base pair fragments cannot be detected because their bands are too faint. And then we tested our primers to test if they would actually amplify the mitochondrial DNA. And as you can see, they're pretty sharp bands. And then we subjected them to the surveyor. And as you can see, because this DNA came from a healthy individual, we have very low levels of heteroplasmy, as shown by the very faint bands marked by the stars here. So the heteroplasmies appeared in the B, D, and E fragments of the DNA that we amplified. And as you can see, the fragments are about 1.2 kb, 600 kb, or 600 BP, 1.1 kb, 0.8 kb, and 1.2 kb. And the other strand for this can't be detected because it's too faint as well. And so we decided to look further into the fragments SCB because those seem to have the brightest bands. And so we found 3 different fragments of DNA that we could amplify that span the whole fragment SCB right here. And we ended up with these results, with some bands found in the 4B fragment, smears found in the 5B, and nothing found in the 6B. So this would tell us that the cleavage we found in the SCB would not be in this region. And then the next step was aligning these fragments, which were 400 base pairs and 300 base pairs, with the previous mismatches that we found in the SCB assay. And so when we aligned them, we found that we could have 2 possible mismatches because of the unknown orientation of the 300 base pair and the 400 base pair fragment. And we have a range at which there could have been a mismatch because the DNA ladders we used for the SCB assay was in increments of 500 base pairs. And because they're about 200 base pairs apart, at least, we're sure that there are 2 different mismatches that we found and not just one. So this proves that the procedure that we developed was efficient in detecting heteroplasmy, and smaller fragments are more sensitive to detecting mismatches. And although we're not sure, we have approximate locations of the mismatches, you would need to sequence the DNA to actually confirm the exact locations of those mismatches. Now, something we found that could have happened with some of the fragments were co-amplification of nuclear DNA, but that's probably not likely because the primers that would have amplified nuclear DNA were found in the C, D, and G fragments, and there are no bands or smears found in the S and G fragments, so we believe that is very unlikely that any nuclear DNA could have been co-amplified. But to eliminate that possibility, we could have separated the mitochondrial DNA from the nuclear DNA before we ran the amplification. And so some future applications of this procedure would be in the detection of threshold levels of heteroplasmy and future research into mitochondrial diseases and the relationship to heteroplasmy. I would like to thank my parents and teachers, Dr. Cecilia Giulivi, Julian Helmai, Stephen Ramsey, Dr. Rick Pomeroy, and all the participants of the UC Davis Young Scholars Program of 2016. Are there any questions?
If mitochondrial DNA is mutated a lot, how often, how many mutations or how long does it usually take for it to become lethal or fatal for the mitochondria?
In most people, it is not lethal, and mitochondrial disease only occurs in 1 every 40,000 people.
Why is it so rare?
Because usually the levels of threshold of heteroplasmy are extremely high, anywhere from 60 to 80%. Yes?
What's the main benefit of using the technique as comparison to, say, classical sequencing?
Okay, so sequencing is expensive and time-consuming, and with our procedure, you can actually determine if there actually is any heteroplasmy to begin with, because with sequencing, you weren't sure if you'd actually get any results or not, and this would save time and money on unnecessary sequencing. Other questions? Okay, thank you for time.
Should we just present or should I wait?
Let's go ahead.
Hello, my name is Matthew Shea, and I go to Amador Valley High School in Pleasanton, California, and I'm working under the supervision of Dr. Cecilia Giulivi and the Department of Molecular Biosciences in the University of California, Davis, and our research concerns a selection of brain biomarkers during the loss of function of palmitoyl transferase ZDHHC13 in mice. Protein palmitoylation is a frequent post-translational modification mediated by palmitoyl acyl transferase, abbreviated PAT, enzymes, a group of cysteine-rich GHHC domain-containing proteins. Palmitoylation increases the lipophilicity of proteins, influencing the subcellular distribution, trafficking, and function. In humans, there are 23 DHHC genes, and in mice, there are 24. Specific to our study, the ZDHHC13 gene encodes for one of the PAT enzymes named HIP14L in both humans and mice. LUCA is a naturally occurring recessive mutation in the ZDHHC13 gene that causes a nonsense base substitution causing a truncated form of the ZDHHC13 protein. Homozygous mice for this mutation experience generalized hypertrichosis and diffuse alopecia. In chemically induced skin carcinogenesis, tumor multiplicity and malignant progression of papillomas were significantly higher in mutant mice than their wild-type littermates, suggesting a protective role for PAT in skin carcinogenesis. In addition, ZDHHC13 is also known as HIP14L, or Huntington interacting protein-like, because it's been shown to interact with Huntington and mice with the truncated form of the ZDHHC13 protein, showing behavioral deficits reminiscent to those of Huntington's disease. Since there's limited information available on the specific biochemical roles of ZDHHC13, we proceeded to perform an untargeted metabolomics analysis. Our goal in this study was to identify potential biomarkers that could be used to define the loss of function of this enzyme. To perform untargeted metabolomics analysis, we used a program called MetaboAnalyst. After uploading various metabolite concentrations from 3 groups of mice on the MetaboAnalyst, the data is then normalized to make the data distribution more similar to a normal distribution. In all of our trials, the normalization procedures were as follows. For sample-specific normalization, we used a normalization by a pooled sample from the control group, the wild-type mice. For data transformation, we used a log transformation, and for scaling, we used auto-scaling. So we mainly focused on 3 procedures in our experiment. Fold change analysis counts the number of compounds with a fold change that are consistently above or below a specified fold change threshold. Partial least squares discriminant analysis, or PLSDA, uses multivariate regression techniques to achieve separation between groups. Each compound is given a variable importance in projection, or VIP score, which basically shows how much of a role that compound had in actually separating the groups. And finally, heat maps are provided as a visual representation of relative concentrations of certain metabolites in each mouse. So for our first trial, untargeted metabolomics in the cerebella of wild-type, heterozygous, and homozygous mutant mice resulted in a PLSDA scores plot that shows pretty clear separation between the 3 groups, with the heterozygous group, as expected, and between the wild-type group and the mutant group. The cross-validation for this plot showed pretty low values for the accuracy, Q squared and R squared, and the permutation test yielded a p-value of 0.8. In addition, a heat map was constructed in accordance to the top 25 components identified by PLSDA. And basically what this shows is that there's fair separation in between the wild-type mice and the mutant mice, with the heterozygous mice showing similarities to both groups. To reduce the noise in the data caused by unidentified metabolites, our next trial included only known metabolites. Surprisingly, in the PLSDA for this trial, there appeared to be more overlap between the 3 groups, suggesting that by cutting back the data, actually less significance was achieved. The cross-validation and permutation tests only had slightly better results. In the heat map constructed, we actually got a full separation between the mutant and the wild-type mice, with the heterozygous mice scattered between both groups. Because neither of these PLSDA models were indicative of a predictive model, we did not consider significant metabolites identified in these trials. Next, we tried looking toward using a more 2-group analysis by combining heterozygous mice with either wild-type or mutant mice in order to take advantage of 2-group analysis procedures such as fold change analysis. In a trial combining heterozygous mice with wild-type mice and comparing them to mutant mice, a fold change analysis with a fold change threshold of 1.3 identified 14 significant compounds. In the PLSDA scores plot for this graph, it actually shows a clear separation between the 2 groups with no overlap whatsoever. And the cross-validation, unfortunately, the cross-validation and the permutation test were still not significant. But with a PLSDA VIP threshold of 0.8, we identified 41 significant compounds, 11 of which were already identified before in the fold change analysis. In the heat map constructed actually shows a full separation between the 2 groups. The only overlaps were due to the heterozygous mice. And in the next trial, we combined heterozygous mice with mutant mice and compared them with wild-type mice. And with a fold change analysis with a fold change threshold of 1.3, we identified 47 significant compounds, 25 of which were already identified through previous statistical procedures. The PLSDA actually shows even a more distinct separation between the two groups, even more so than in the previous trial, suggesting that heterozygous mice may be more similar to the mutant mice than to the wild-type mice. And the cross-validation was actually the best out of all three groups, and it showed not too much improvement, but it was still the best among them. But unfortunately, the permutation test was still insignificant. With a PLSDA VIP threshold of 0.8, we identified 47 significant compounds, 41 of which were already identified through previous statistical procedures. And in the heat map, it actually shows a fair separation between the mutant mice and the wild-type mice, with heterozygous mice actually showing more similarities to the mutant mice than to the wild-type mice. So overall, untargeted metabolomics resulted in the detection of 72 total unique metabolites, and mutant mice showed differences in 44 of these unique compounds. Of these compounds, cysteine appears to be an appropriate one to focus on. It is found in beta-carotene and shows an importance in collagen production, as well as skin elasticity and texture. And PAT enzymes also contain a large concentration of cysteine, so it's expected that cysteine should be identified as significant. Taurine should also be looked into further because the synthesis of taurine involves both cysteine and B6. So if cysteine is identified as significant, taurine should be too. Oxyproline, a cyclized derivative of glutamic acid, may also be important because it is found in large amounts of bound tissue, such as skin. And 2-hydroxyglutaric acid, a known oncometabolite, may have a role in the actual progression of skin cancer in the mice. To differentiate wild-type mice from the rest of the group, we identified 59 unique compounds. And many of the metabolites identified in the previous group, such as cysteine, taurine, and oxyproline, were actually also identified as significant in separating wild-type mice from the rest of their group. And possibly further research may involve tracking specific metabolites throughout the pathways of the mice to find possible roles that each of these compounds have in the actual progression of skin carcinogenesis. In the PLSDA VIP graphs for the last two trials, it appears that a vast majority of the fatty acids identified were found in high concentrations in wild-type mice and low concentrations in mutant mice, possibly suggesting a downregulation of fatty acid synthesis, or more simply, just a higher use of fatty acids as a consequence of the ZDHHC13 mutation. For compounds involved in cellular respiration, a majority were found in low concentrations in wild-type mice and higher concentrations in mutant mice, possibly highlighting differences in mitochondrial function. Concentrations of amino acids, sugars and sugar derivatives, nucleic acids and nucleotides are shown in the table above. And heterozygous mice consistently laid somewhere in between the wild-type and the mutant mice. By grouping these specific metabolites into more general groups, we can better determine the role of HIP14L in more general metabolic pathways, and in addition, we can get a better overview of classes of molecules rather than being restricted to focusing on each individual metabolite, allowing us to predict the role of other compounds possibly in these mice later on. By identifying potential biomarkers, this research could lead to the development of better identification techniques leading to earlier detection and possibly earlier treatment. Further studies should include larger samples of mice because a larger sample size would mitigate the effects of data variability on the results. The very small sample of mice in our experiment appeared to show limited outcomes in some of the areas, especially in the PLSDA cross-validation and permutation tests. Further research could lead to a better understanding of the mechanisms relating to ZDHHC13 mutation to skin cancer itself. First off, I'd like to thank my parents, Mom and Dad, for always encouraging me and allowing me to pursue opportunities like these. And I'd also like to thank my professor, Dr. Cecilia Giolitti. I know I wasn't always the best young scholar in lab. I'm sorry if I ever caused you any trouble. This has been an experience and a lesson that I'll keep forever with me, and you're directly responsible for that, so thank you. And I'd also like to thank my counselors, Stephen Ramsey and Dr. Rick Pomeroy, the director of the Young Scholars Program, as well as all the participants of the Young Scholars Program, and all of you sitting in front of me today. Thank you.
What is hypertrichosis and alopecia?
Hypertrichosis is a skin condition which causes enlarged, basically skin cell layers. And alopecia is the loss of hair. So if you see bald people with no eyebrows, that's alopecia. Brian?
So if the PLSDA validation was not significant, why did you still analyze the significant compounds?
So for the last two trials, we also had the voltage analysis, which analyzed significant compounds. So for the PLSDA, we decided to try to compare those two metabolites to see if they had any overlap. And also the cross-validation for the last two trials of PLSDA were the best that they've been throughout the trials. And the permutation graph, as I said before, was probably affected by the small sample size. So we pretty much disregarded that in identifying the metabolites.
So what is the role of metabolites in cancer? Or is there a relation between
So
metabolites?
that's basically what we're trying to find with this experiment, finding the possible metabolites that can be used that play a role in the development of skin carcinogenesis. But mainly, the metabolites are involved in finding the link between a lack of PAT enzymes in skin carcinogenesis, which is basically what we're trying to find in possible further studies.
You had some molecules today in the discussion.
So we have cysteine. Do you want to read this off?
So what were how were heat maps used in identifying metabolites? Or what are heat maps measuring?
They're actually not used in identifying metabolites. It's more in showing similarities and group separation. So actually, for example, better visualization will be the 3 group ones. So here, if you see the wild type mice, a lot of the compounds here are shown in lower concentrations. So it basically just shows the similarities of concentrations of these metabolites within the groups. So basically it provides a visual representation, and you try to see the colors match up to try to group them together in the different groups. So as you can see, some of the heterozygous mice, they show blue streaks here, similar to the wild type mice, but some of them also show red streaks here, similar to the mutant mice.
So these are concentrations in metabolites?
Yeah, relative concentrations. So higher concentrations are darker red, and lower concentrations are dark blue, basically.
Thank you.
Up next, I'd like to introduce my colleague, Jackie Ho.
We're running about 5 minutes early. Is everybody who's going to be here?
My parents are going to be watching at home.
Okay, then we're going to wait.
Okay.
So we'll give 5 minutes so that we don't miss them, okay?
Unless you want to call them and tell them to turn on their computers. Okay, I'm gonna go.
Okay, hi everybody, my name is Jackie Ho. I'm working under the instruction of Dr. Madhu Buddhamagunta and Dr. John Voss at the Voss Lab, which is in the Department of Biochemistry and Molecular Medicine at UC Davis. So my project has two main parts, the first of which is the comparison of the apolipoprotein E4 and its 61T mutation, and the second which is the lipid bounceability of apolipoproteins in general. So that complex title essentially boils down to two main things, which are apolipoproteins and their role in the development of Alzheimer's. So Alzheimer's is a neurodegenerative disease. It's characterized by the breakdown of neurons in the cortex. What happens is that amyloid beta, a protein that's naturally found in the membranes of neurons, begins to oligomerize, and it forms plaques. These plaques tend to clog up the synapses in between neurons. The result is that they can't form connections, which are fundamental to making memories. So with early stages of Alzheimer's, you see patients that are unable to remember things like what they had for breakfast or like where they are right now. But eventually, this becomes so severe that they cannot remember the most fundamental things about who they are. So the reason why research into Alzheimer's is so relevant is because in the US alone, there have been 5 million people diagnosed with Alzheimer's. And this amounts to approximately 8.1 billion hours of unpaid care. And for each patient, it's estimated that care requires approximately $5,000 per year. Now, for some of us, that might be affordable, but for many others, this kind of time and money is not something that they can afford. In addition, it's approximated that every 66 seconds, another person in the US alone is diagnosed with Alzheimer's. Now, the other main part of my project is apolipoproteins. Now, if you dissect the word into its roots, you have apo, which means to carry, lipo, which refers to lipids or fats, and proteins. So it defines itself. It's pretty much a protein that carries lipids or cholesterol-like molecules. The apolipoprotein E is the one involved in Alzheimer's. There's 5 main types. And of the ApoE proteins, the most common is the E3 protein, which is found in the majority, or approximately 70% of the population. And the one found in Alzheimer's patients is the ApoE4 protein. Now, if you look closely, they only differ in a single amino acid, which is right here at the 112th spot. In E3, it's a cysteine, which is small and uncharged, while at E4, it's an arginine, which has a positive charge. Now, this makes a huge difference in the overall structure of the protein, because as you can see, the 112 is among the alpha helices here, but on this side chain, on the carboxyl terminal, you have a glutamic acid, which has a negative charge. And what you can predict is that the arginine in the E4 found in Alzheimer's, it interacts with the glutamic acid and brings the structure together and makes it much more condensed, so the protein cannot carry amyloid beta out of the brain. And as a result, you get these plaques in the brain which cause Alzheimer's. Now, in my project, we examined the effects of 6 drugs on the E4 protein, which is the normal found in Alzheimer's, and a mutant we made and called 61T. Now, to determine whether the drugs had any different effects on the 2 proteins, we used electron paramagnetic resonance spectroscopy, or EPR. And how it works is that a strong magnetic field is created with a detector in it that senses the movements of lone electrons. So how it works is we attach spin labels to the cysteine amino acids throughout the protein, and then we measure the movements of the lone electrons to judge the surrounding structure. So here I have 2 diagrams of spectra that we received. So out of the drugs A through F, their identities must remain confidential because they're still under development. But we have spectra that are very different here for drugs B and F. So you can see there's a clear change in amplitude as well as shape, which indicates that for the 61T mutation, it reacted differently than the normal clone S. Now, while these amplitude changes seem pretty significant in this diagram, in repeat studies, we had inconclusive results. For example, in dilutions and a repeat scan of all the drugs, about approximately 2 weeks later, we only found a big difference in drug F. And this may indicate that the protein may have degraded over time, or it was calibrated weird in the first run, but we have to repeat scans in the future to determine like if the results were actually significant. And now I'm going to transition to the second part of my project, which regards the apolipoprotein A1. Here we have diagrams of previous research on this protein. How it works is it binds to lipids to form like a tiny hockey puck, and you have these proteins that surround a circle region of lipid bilayers. These are the same lipid bilayers that you would find in a cell membrane. And because of that, these kinds of nanodiscs are super popular in biotechnology, because with these, researchers can work with integral or transmembrane proteins and effectively stitch these pieces back into the cell membrane. So here we have a diagram of a Kumasi assay and an SDS page gel of 8 samples. As you can see, 4 of them are lipid-bound and 4 of them are lipid-free. Now this was unintentional, but we found that the lipid-free samples that we obtained were all at higher concentrations, so they appear more blue in this assay. And the lipid-bound are brown or lower concentration. And this also reflects in the SDS page gel, which is essentially a protein gel. You can see very faint bands on this side and more dark bands on that side. Now, another thing we noticed in this gel is that while you have these two, specifically Y30 and L183C, which have very smeared bands, in the lipid bound, although these are faint, they tend to have more clear and distinct bands. So it might indicate that the structure of the protein remained more stable over time. Because all of these samples were taken from previous research, so some of them date anywhere from like 2003 to 2011, so these are fairly old. So we ran scans on 4 of the lipid-bound and lipid-free samples. These are the 2 lipid-bound samples, 170 and 171. And right away you can notice a big difference between this and this one here. So with the 170 lipid-bound, you can see that these peaks are pretty sharp before and after adding SDS. And SDS is essentially a detergent that breaks up the protein and opens the structure. So here you can see those peaks are so sharp, which indicate that the spin labels were already free and moving a lot before adding SDS and after SDS. But in this side, you have the 171, and you can see clearly that before adding SDS, which is black, you don't have these sharp peaks, and it's much more broad and asymmetric, which indicates that the spin label and the lone electron was jammed inside 2 things, or like surrounding structures, so the electron didn't move as much. And you can see after we added SDS to open up the protein, you have these much cleaner and symmetric spectra. And here is a spectra of the lipid-free protein. And this is what we typically predict from seeing samples like this. You have these clear, sharp spectra before and after adding SDS, which indicates the lipid-free had already been opened to some extent before. So another point that I failed to mention earlier is that the 170 and 170 nanodiscs, they differ in diameter, which is another crucial part. The 170 was 7.8 nanometer lipid bound, which means it was slightly smaller, and the 171 was a 9.6. So as you can see from these SDS page gels, the 9.6 tend to have darker, more concentrated bands, even though they were stored at the same concentration as some of these 7.8 samples. So what that might indicate is that the 9.6 nanometer lipid-bound samples retain their integrity better than the 7.8 nanometer lipid-bound, which is fairly consistent with previous research. So that might be the explanation why the 170 spectra looked so similar to each other, like they both degraded because they were 7.8, whereas the 171, which is a 9.6, had a bigger difference, which indicates that the lipid-bound structure was still intact. So in conclusion, over this short time frame, we weren't able to run very many scans. So in the future, we would like to conduct more scans to see if the drugs D and F were actually effectual. And for the second part of the project, we would want to have more spectra because we used different diameter nanodiscs and they were of different ages. And there's so many more things we can control for to determine whether the results were actually significant. So I'd like to thank Voss Labs, specifically Dr. John Voss, Dr. Madhu Brahmagunsa, and my grad student Sylvia Hilt, and everybody in the UCD YSP program, specifically Dr. Pomeroy, Karen Rushing, and my best roommate Renee, and all of you guys.
Earlier in the beginning of the slide, we showed the difference between like ApoE83 and ApoE4 was like a difference in a single amino acid.
Yeah.
Are there other mutations where
So there's 3 alleles of the ApoE protein. There's V2, which is actually very rare, and it actually is associated with like increased risk of vascular disease, so that's why we don't really want to consider it. And then there's the E3 and the E4. So they all differ at like a single amino acid.
Any other questions?
They're hungry.
Thank you. All right, ladies and gents, we will be back for the second half of the day. Please be here about
1:25. Yeah. Oh, you're welcome. Good job. Good job. I gotta know what you did. Celia looked at me and just laughed. So I'm sure it wasn't critical because she never called me. Okay? Good job. Nicely done.
That's what I said, yeah. I said
Very good job.
Yeah. I got your fair results for your No, that was good. Show me the other one.
I want them returning their bikes now.
I just want to see your viewpoint compared to mine. Because I thought that was pretty cool.
Do the bikes
have to be turned in today?
Yes.
They must be turned in today?
Yes. About the 5th time I've said that. Didn't really need you to ask me twice more.
Henry, run away.
Go.
Henry, run away.
Just go. Go return your bike.
Do you see? Or somewhere else?
Okay.
Somebody lose this?
They'll figure it out in just a minute.
Here, I'll take it.
Here, we'll take it. Because they won't be going very far. Okay.
Yeah, and Rick has a power cord here.
Could I go over to the bike barn?
That would probably be a good idea. Just so that, yeah.
You wanna walk to the bike barn?
If you'd like to, that's fine. Leave
I'd
us,
say we're the two bike.
leave us the keys to the car.
Actually, Annie.
You guys are going to the bike barn. We'll take, where's the park, by the way?
Parked over in the library.
Art annex?
Yeah, by the art place, yeah. Or by the art building.
Yeah.
Okay.
What? Or you can take it, but here, I'm parked over there too.
Yeah, right back when you're done.
So if you want to drive back after the bike barn.
That'll work. Did you put Carter's sign right over here?
Yeah. I was going to take the computers.
Yeah.
Oh, shoot. The world's listening to us.
Hello, world. Okay, I'm gonna run to the restroom.
Yeah, the world's
Ben, are you missing something?
Yeah.